Hello and welcome back to another week of histological techniques at QUT. This week we've been studying mucosubstances, and it is a technique, or a topic I should say, that I've covered on this channel before by looking at the PAS staining technique. In this video, and it's been quite a challenging video to make because of the complexity, just deciding how much detail to go into and how to keep it as simple as possible. In short, what we've covered previously in looking at the PAS technique is a relatively simple method for demonstrating the presence of carbohydrate, mucous substance, in tissues, which has a neutral chemistry. And I'll define what I mean by that in a moment. This semester, however, in the studies that have been conducted here at QUT for the students, the undergrad students, we delve a lot more deeper by exploring the complexity of saccharide chemistry. That is to say that not everything can be considered neutral. So the options are for things to either be neutral or they could be acidic. That is, under physiological conditions, certain types of mucosubstances have a predominance of negative charges. Okay, so in terms of histochemistry, things that are negatively charged can be said to be behaving like an acid because they're giving up hydrogen ions. Okay. So let's begin by looking at this stained slide of colon. And in this section, we have applied not only the PAS stain, which you can see evidence of PAS staining by virtue of this bright pink staining here in the scoblet cell. And there's various levels of pink staining within other structures. Even the collagen to some extent has got a little bit of color. There's a bit of staining around these blood vessels where there are smooth muscle cells or pericytes, etc. But we also notice there is a bright, vibrant blue color. And that's because we've employed a second dye called Austrian blue. Simply put, Austrian blue is a positively charged dye which stains negatively charged mucous substances, or mucous substances which are termed to be acidic. Okay, so it makes a nice combination of staining with the PAS technique, which, as some of you may know already from my previous videos, the PAS staining technique is effectively staining those saccharides that can be considered to have neutral chemistry. That is, there is no negative charge within a particular saccharide group. There are just hydroxyl groups, and in particular, there are two hydroxyl groups located adjacent to each other, which when oxidized form a dialdehyde, which subsequently reacts with Schiff's reagent, which then when you wash well with water, creates a conformational change within the Schiff's reagent such that it loses a sulfonic acid group, and then you generate this lovely pink color. Okay, so that's what's been done to generate this particular slide here, stained of colon, where we can see the pink and the blue stains working together. So what I thought I would do in this first video, first of two this week, is to look at some of the chemical structures which are responsible for leading to these variations in staining properties of mucous substances. And then in the second video that I'll also release today, we've got some really nice sections through the digestive tract, starting with the tongue, working our way through to the large intestine and even some rectum. And by applying the PAS and the Austrian blue stain, we get a better feel for how there is variation in the composition of these acidic and neutral mucosubstances as we move our way through the length of the digestive tract. Okay, so turning our attention now back to the screen here, I'm just going to share the online course. This is similar to the one that we use here at QUT, but this is the one that's made available to those who join the channel as a trainee histologist. And if we make our way down to lesson 9 here, this lesson actually doesn't contain too much in the way of images. There's a Movats pentachrome image at the start here, which I'll have a separate video on that in a few weeks' time, hopefully. But really the benefit of sharing this information is that it provides some structures that I've created in ChemDraw as a way to provide a foundation for an understanding of the differences in the histochemistry of different mucosubstances. Okay, so if we begin with the simplest of structures, and admittedly, for those of you who may not have done much chemistry previously, this is as complex as you would want it to be, but just bear with me. So this represents a generic structure of the simplest subunit that you would find within carbohydrates, sugars, mucosubstances. I'm using those terms collectively. In a histological sense, we refer to carbohydrate present in tissues as mucosubstances. But chemists refer to this as a saccharide. In fact, it's drawn as a monosaccharide. This is actually the chemical structure of glucose. And through this layer here, these various joining black lines, this represents a ring of carbons with an oxygen atom over on one side. And most significantly, you'll note that there are these OH or hydroxyl groups located in various locations. This is significant. These two hydroxyl groups located in these positions bestow the property of making mucosubstances found in tissues that have this type of structure. It bestows upon them the ability to be stained using the PAS staining technique. So if you've seen the other video on PAS staining technique, you'll recall that through oxidation of these adjacent hydroxyl groups, this is where we get the formation of the dihaldehyde group, and then subsequently the reaction with Schiff's reagent, and then subsequently with the rinsing in water, we generate colour. Okay, so that's fairly straightforward. Importantly, you'll note that there's only really hydroxyl groups and hydrogen groups, which are the functional groups found within this saccharide ring structure. Let's now compare that to this next figure, where again, we've got glucose there again on the left-hand side, but on the right-hand side, we have a saccharide structure which looks quite similar to what we have on the left, but this is actually referred to as beta-D-glucuronic acid. And you get a hint from the name there, it's acidic. Why is it acidic? Because this structure here, the carbon with two oxygens and a hydrogen, is what we call a carboxylic acid. And under physiological conditions of pH, say relatively neutral pH, I guess, we will find that this structure wants to give up a H+ hydrogen ion. That is to say, it wants to behave as an acid. And in doing so, what's left behind is a negatively charged group. So when we talk about negative charges in histochemistry, we are talking about structures that are behaving as acids. Think of nucleic acids and the previous videos that I've made about staining with hematoxylin. The positively charged complex of hematoxylin and the aluminium mordant, which is commonly used, will bind to the negatively charged nucleic acid within the chromatin. And so we'll see nuclear staining. So this is a rather similar principle. These negatively charged groups on these saccharides therefore make them a candidate for staining with positively charged dyes. And in fact, in the second video, we'll start by looking at a H&E stain section. And you'll actually see that there is some degree of staining for mucosubstances from the hematoxylin. Okay, let's move on to a more complex structure. This time we have glucose, but it's glucose in the form in which it's normally stored within our tissues. So certain types of skeletal muscle fiber and liver are often places where we will find varying amounts of glycogen being stored. Glycogen is a polymer of glucose. It's basically our reserve. It's our way of storing the glucose until it is needed by the body. And so that's represented here as this series of glucose saccharide monomers, which are linked together by these glycosidic linkages. Once again, you can see that as a consequence of being polymerized, these glucose monomers have avoided generating any negative charges. And so this, what we call polysaccharide of glucose or glycogen, will still retain similar staining properties to what glucose has. And so in fact, when we're talking about staining for glucose in tissues, we're not usually staining glucose, we are actually staining glycogen. And so we'll see some examples of staining for glycogen in skeletal muscle in the second video. Okay, so absence of negative charges, but we've got these adjacent hydroxyl groups, and so they'll have the potential to be oxidized to a dialdehyde, and then will subsequently react with Schiff's reagent. Okay, so we are aware that there are neutral saccharides like glycogen, and there are acidic saccharides. Well, where are these acidic saccharides found? They're not in glycogen, so where do we find them? Well, there's two areas where you can find them, and we generically refer to these as glycoproteins or proteoglycans. They are, at their core, very similar. It's a combination of protein and carbohydrate. How they tend to differ is in terms of their size and also their relative composition of very acidic mucosubstances. So if we start by looking at glycoproteins, glycoproteins is a very broad family of proteins found within the human body. Essentially, many proteins, as part of their post-translational modification, will have varying amounts of oligosaccharides, that is, short chains of carbohydrate, which will be attached to those structures, and it's often part of their normal function that they need to have that saccharide there. Now, if there is an absence or a low proportion of acidic saccharide groups within those chains, well, then these glycoproteins will be predominantly neutral in their chemistry, and they will stain up using the PAS stain. However, if they obviously have more of those acidic type of saccharides, then there is potential to be stained with Austrian blue. If we take that concept further, proteoglycans are really a separate category based upon the fact that these are a very specialized family of glycoproteins found in the extracellular matrix, that is the environment outside of cells. Sometimes they're stored, or components of them can be stored internally, but proteoglycans are generally very high molecular weight molecules consisting of a protein core and very long chains of saccharides, which are special chains known as glycosaminoglycans. And part of the reason why you can distinguish proteoglycans from just a generic class of glycoprotein is that glycosaminoglycans that form these long side chains have many highly acidic motifs within their structure. That is, if we look at the saccharide subunits that make up those side chains, there's lots of negative charges. And it turns out that not only are they negatively charged, but their ability to become negatively charged is a lot stronger than the structures that we've already seen, which were carboxylated. Let's go down and look at another structure, and this will explain where this highly acidic structure comes from. Okay, so as we move to this next figure, what we can see, first of all, on the left, we have a series of saccharide rings where there is once again these hydroxyl groups which are present, but there's no presence of carboxylic acid groups, as we've seen previously. On the right-hand side, however, we see a more complex structure, and as you can tell from the name, there are sulfate groups which are present within these structures. Now, we haven't mentioned sulfate yet, but it turns out that sulfate groups, like carboxylate groups or carboxylic acid groups, are actually even stronger acids. That is, their ability, their willingness to give up hydrogen ions to the surrounding environment is even stronger. Okay, so what does that mean? Well, that means that there's actually at least two levels of acidity for acidic mucosubstances, some which are weakly acidic, and there are some that are strongly acidic. And it's all by virtue of the relative composition of the amount of sulphated saccharide subunits which are present. Now, moving on to this final figure, and my apologies, this has been quite a complex journey through all these structures, but hopefully you'll have the benefit of this when we go to the next video. We'll be able to apply this terminology a lot more freely when referring to various structures that we can see down the microscope. So this final structure is an example of a sialic acid. Again, it's just another type of saccharide. We can see the typical ring-like structure of carbon and oxygen, but the arrangement of side groups is far more complex. This has a mixture of chemistries. If you look, first of all, to try to find the presence of adjoining hydroxyl groups, we note that within the saccharide ring structure, there are none to be found if you look closely. However, we do have this side chain going down here at the bottom of the image, and within that, we have adjacent hydroxyl groups, some of which can be very effectively oxidized using periodic acid to a dialdehyde and then subsequently stained with Schiss reagent to generate that lovely pink color. But at the same time, we've also got a carboxylic acid group over on the side here as well. So these are generally regarded as weakly acidic, but there will also be some reactivity towards PAS as well. Okay, so there you have it. That's quite a complex series of images, and I'm hoping that that will provide a foundation as we move to the next video in a few moments' time. But just to summarize then, what we're actually looking at within tissues, when we are staining histochemically for mucosubstances, there's obviously more sophisticated ways using immunohistochemistry, but using dyes such as PAS and Austrian blue, the four categories that we're actually able to distinguish between is there'll be neutral saccharides with those glycol groups, that's the name we give to those adjacent hydroxyl groups, which can be stained up using PAS. There will be weakly acidic saccharides with carboxyl groups. And there may be others, such as the sialic acids, which are weakly acidic, but will also have some reactivity towards PAS. And then in the case of proteoglycans that we briefly discussed, those by virtue of their sulfate groups will have very strongly acidic saccharides present. Okay, so thanks for your attention. That's the foundation. When we come back, we are going to apply all that knowledge using not only the PAS stain and Austrian blue, but also a third dye called mucicamine. And we'll apply that knowledge to take a journey down the length of the digestive tract, and we'll see how the relative composition of acidic and neutral mucosubstances changes as you move from one area to the next. Okay, so that's enough for now, and I'll see you very shortly. Bye for now.